life continuing in that way. I mean, we do have the ability to have children, right? Most of us can do that eventually. And so in a way, your offspring can live on, but really it's not the same, right? Because in that scenario, the parent doesn't become the child, right? And that's what we're talking about here today, is the parent cell becomes the child cell, the daughter cell. Okay, so cell development. Cell development has two parts. When we think about cell development, that's just a fancy way of saying the life of the cell or how the cell grows. Okay, so development just means how the cell grows, how it goes from being an itty-bitty baby cell all the way to a full-grown adult cell. Okay, so you can think of the stages that a cell goes through in terms of growth as having two parts. So let's just number these two parts. We'll underline the names of the parts, okay? So the first part is called interphase. Okay? And this is actually where the majority of the life of the cell is, right? You guys have different phases of growth too, right? You guys were babies at one time, right? And then you were, children, right? Then you were maybe teenagers or whatever that is. And now you're just full teenagers, right? You're adolescent teenagers. And soon you're going to be adults, right? You're going to be living on your own, driving your own car, doing your own job, having your own stresses, getting your own coffee, right? And then eventually you're going to get old like me, and you're going to start falling. Hopefully not. But that's interface for the cell, right? All of that is interface. The cell grows and grows and grows. And so what's the cell doing? Is it just hanging out? No. The cell has a job, right? It's doing its job. And different cells have different jobs. We know this, right? There's muscle cells. Muscle cells have an important job, right? They allow us to move. There's bone cells. Bone cells have an important job. They protect us, right? Our rib cage protects our lungs. Our skull protects our brain. And our bones also allow us to move in addition to that. So our cells have jobs to do. So as they're growing, they're doing their job inside of us. Okay? It's really important that they be able to do that job because that's what keeps us healthy. But eventually, a cell's going to get big. Okay? And when it gets big, remember, it has multiple options. It can stop growing. Or what happens in a lot of cases, and is certainly happening in your bodies because you're still growing up, is it divides. And so when the cell divides, there's two things that have to happen. The first thing is that the nucleus needs to divide. Let's underline the letter M for mitosis, okay? You've heard of mitosis before. A lot of people make the mistake of thinking mitosis is cell division. It's not. It's just when the nucleus divides. And then there's a separate part called cytokinesis. Let's underline the C for cytokinesis. That's when the cell actually divides, okay? The cell membrane divides and everything in it, okay? So here's this diagram, or this circle here, shows us that this process is actually a cycle, right? This process is actually a cycle. Let's think of it as starting right here, okay, as little bitty cells, okay, little tiny baby cells. And those cells go through a bunch of stages of development until eventually they are big. Now, big is relative, right? They're not as big as us, but they're big for a cell. And so that's when the cell decides that it's going to need to divide. And so let's label a couple of things here. First of all, right here, let's label this I for interphase. I for interphase, okay? Interphase, remember, that's when the cell is doing its job, it's growing, it's getting bigger, and it's performing a function inside of us. And then eventually, the cell is going to start to divide, right? So let's put division here, D for division. But let's label the first 4 parts of division. Mitosis with an M. And number 5, and just 5, we're going to label C. Okay, so that's why we underline M and C. Okay. I for interface. I for interface. Yeah, shoot. We didn't. Maybe we underlined the whole word. Maybe I'll circle the I. T there. Okay. So maybe we can also label it too. We should do that. Abbreviations sometimes don't save time, but these ones save space. Okay. So interface, right, and division. Those are the two parts of the life of the cell. Okay. Yes. Sure. Yeah. This is exactly that. Yeah, you got it. The question was for those of you that did hear is this anything like asexual reproduction? Asexual reproduction is when you just have one parent, and essentially the offspring are clones of the original parent. So yeah, that's exactly what we're talking about. Our bodies do asexual reproduction all the time. It's how we grow. It's how we get bigger. It's how we repair ourselves when we get injured. Okay. So you might have noticed here the interface has a bunch of little parts. Okay, so let's go ahead and label this. Label these different parts. By the way, I don't want to jump over this. Do you see how this is a cycle? Right. Once the cell gets big and divides into small cells, then the next cell is going to go through this phase and get big and divide, and then they're going to go through this again. So it's just a constant cycle, right? The cells don't necessarily die; they just get big and divide. Okay. All right, well, let's take a look here. There's multiple parts to these. Interface, we're going to label gap 1, or if you want to use the abbreviation, which is dangerous, G1. G1. Okay, sometimes we abbreviate it G1 for gap 1. Okay. This is the part of interface where the cell's growing and it's doing its job. For each part of the cell cycle, I want to focus us on what's the DNA. Since that's what we were just talking about, and how DNA codes for proteins, the tools that make us work. For each of these parts, we're going to think about how is the DNA being used. Oh, okay, there you go. So how's the DNA being used? So in gap 1, the DNA is being used to make proteins needed for the cell to do its job. So remember, we just talked about that. Let's make a connection to what we just talked about, transcription. And anybody remember the second one? Yay, great job. Translation. So that's when the cell is doing transcription and translation, it's doing that during gap 1. Right? So what we just talked about is happening during this first part while the cell is making all of the tools that it needs in order to do its job. The second part, we're going to abbreviate with an S. S stands for synthesis. So synthesis is the next part of interface. And what are we synthesizing? We're making more DNA. This is DNA replication, which you guys remember 2 units ago. That's what we were talking about, how DNA can unzip. Free-floating nucleotides can base pair, G with C, A with T. And then DNA polymerase, the enzyme, comes along and makes a new DNA molecule. Okay, and so let's see, where do we want to circle here? Let's just circle the word replication. This is when DNA is doing replication. And I don't want to leave this spot because I'm going to lose all my annotations. But if you keep reading down below there, right? Actually, I'm going to have to. I'm going to do this. I won't lose them. I just want to point this picture out, right? So if we take a look here, right, we see in a cell 2 DNA molecules, right? After DNA replication, they look like this. It kind of looks like the letter X, right? But what we're saying is each of these halves of the X are identical to each other, okay? And we call those sister chromatids. So even if you wanted to label where it says capital A, label those there or draw arrows from there to sister chromatids. I guess I can do that. It's going to be a little bit weird. I'll use ignore all the red, okay? Sister chromatid, there's a sister chromatid, there's a sister chromatid. Those are identical chromosomes that were copied from the DNA, the original. Okay. for all the red. We'll go back to the red here in just a moment, okay? Okay, so let's see. I get this to line up again. Okay, here it is. Okay. And now, all right. So the next part of this is gap 2. Gap 2, which is not a very original name, right? But it's after gap 1. Sometimes we abbreviate that one G2. Okay. In gap 2, the cell is doing more protein synthesis using its DNA, but this time it's making tools that it's going to need to divide. And so for every cell that divides, we have the same tools. Okay, so these are specialty tools. I think of these as like the saws, right? Because you can imagine, not to be too graphic, right? But maybe there's going to be a saw involved in cutting these cells into 2 cells, right? They're going to need special tools for dividing the cell into 2. And so we've got to make those during gap 2. Okay? So think of this as kind of like preparation for cell division. Okay? Any questions? What we're going to do next is look at mitosis and cytokinesis, because this is where there's lots going on that we can visualize and think about. So I'm going to go ahead and erase this. Does anybody need more time with the annotation? You took a screenshot. That's a great way to do it. If I'm going too fast, just take a screenshot and you can add it later. Oh, good question. I did just erase that, didn't I? Cytokinesis would be just number 5. Yep. And 1 through 4, that would be mitosis. Okay.
Mr. Kramer, one more thing. What if you have problems growing?
What's that?
Problems growing.
Are we talking about cells or people?
People.
People, yeah. Sometimes that can happen. Usually that's a hormone issue.
Yeah.
So hormones control the way our cells behave and act. And so that could affect, how that person's cells divide and how quickly that happens for them.
Yeah, I was one of them. Same with my sister.
Okay. I'm glad you're with us and doing so well, Jenna. Okay. I can't wait to meet you. Someday we're going to meet. It's going to be amazing. I mean, I feel like I know you, but I've never actually seen you. I've never actually had you in my class. I can't wait for that. Someday you'll come by, right? Maybe once COVID goes away. Okay, so gap 2, let's see, gap 2, the cell prepares for cell division, right? The cell prepares for cell division. The DNA is used to make proteins needed for cell division. If it helps, let's think of like a saw, right? It'd be proteins that are going to be needed to divide the cell. And so this is transcription and translation again, but this time for the purpose of one job, which is the big job, dividing the cell into 2 parts, okay? Any questions? How we doing? I feel like you guys could use some coffee too. Let's look at the parts of division that will be done for today with any new material. We'll just practice what we learned here, okay? So the first part of cell division is mitosis, and that's where the nucleus divides. And of course, you guys know what lives in the nucleus? The brain, good guess. We think of the nucleus as kind of like the brain of our bodies, but the nucleus is just a part of a cell, and the brain is made of trillions of cells. What's that important molecule that lives inside the DNA? Where's DJ? It's the DNA, right? The DNA is inside the nucleus, and so we're going to need to be able to get these sister chromatids to separate so that they can go into each daughter cell. And so this is going to happen in a couple of steps. The first one is going to be prophase, and we're going to let's underline the P for prophase because that's how we'll abbreviate it, okay? Prophase. And if you're a professional athlete, right, pro means first, right? A professional athlete is a first-class athlete, right? They don't have a side job, right? Their job is to be an athlete. So pro means first, okay? This is the first phase of mitosis, and this is where the DNA lines itself up into chromosomes, okay? Chromosomes are just DNA that have been balled up into a really short version of itself. DNA, we said, is a super long molecule, and I always think of it this way: if I have a long piece of string, say I give you a mile of string, okay, and I say this is my prize string. It's really important to me that this string be protected and that this string not fall apart or get frayed on the edges or lose any pieces. That would be awful. What would be the first thing that you would do to protect that string? I gave you one end of it, and the other end of it's down at the gym. What would you do with that string to protect it? Would you just leave it all floppy like that and just drag it around the school all day? People stepping on my string, my favorite string. No, you wouldn't do that, would you? What would you do? What would be a more efficient way to carry a big, long piece of string? Yeah, roll it up. What'd you say it's called? Yeah, get the other side. At least that would, divide it in half, right? And yeah, you could roll it up, right? You could ball it around your hand, right? And have a basketball of string that you could easily tuck into your backpack and carry it around that way. That's basically what a chromosome is the DNA is in string form. Okay, it's really long and skinny and vulnerable to breaking apart. But when we wind it up into a short little ball of DNA, we call that a chromosome. Okay, so think of a chromosome as it's like a ball of DNA. That's all it is. Of course, it's not totally round, is it? It's elongated, but that's basically what it is. And so what this picture is trying to illustrate is that the DNA goes from being a double helix over here on the right. That's how we think of DNA, right? It's a double helix. But it wraps around. This is called spooling. It wraps around proteins called histones, and then they start to twist on each other, and they supercoil until eventually you have a chromosome. So chromosome is just DNA that coils up on itself. Sure, this one? Yeah, it's not doing anything. Okay? And so a chromosome is a ball of DNA. So that's what I tried to show here in our illustration here. I made these drawings just to make them super simple here. Let's just identify a couple of key things here, okay? First of all, the DNA is winding up. It's shortening into chromosomes. So if you want to, let's draw an arrow, I should say. You see how I made it look like a string that's coiling up? Okay, that's a good way to think about a chromosome. The second thing that's going to happen during prophase is the nucleus is going to fall apart. So I showed it this way. The nucleus, I drew it as a dotted line. And we don't actually understand how this works, but the nucleus actually falls apart. And not the cell membrane, which is fascinating to me to think about, because if the cell membrane fell apart, then the cell would be dead. So just the nucleus falls apart. And then the last little bit of this, let's draw an arrow going up to here. These are called spindle fibers. So the spindle fibers are strings. We're going to think of them as like fishing line. And they start to form in the cell as well. They're going to use those later on. So three things happen. The DNA is winding up into chromosomes, so it becomes shorter and safer. The nucleus starts to fall apart so that the DNA can get out. And then the spindle fibers start to form. This is like fishing. Okay. Any questions? Everybody draw the arrows? We're getting there. Yes. We're good? Yes. Absolutely. Do you want to do that in the bathroom? Or do you want to use our sinks? That one's big enough you could climb in. If you want to just douse your whole body, we've got 3 spigots going. Okay, no problem. Yeah. Should we take a break? Can you pause, or is that not possible? That might not be possible. We're going to keep going. Yeah. All right, I'm going to keep going here. So the first part is prophase. Now let's go to metaphase. This is page 2, okay? a dance. Okay, it's like the chromosomes are doing a dance. They follow the same pattern when they do this. So it's all about the pattern.
Like that, like going at and then displaying part time.
Yeah, you're right.
Like as they get hotter, they speed up and then they go out of control.
Wow, yeah. And this is predictable, okay, just like that. So metaphase, when we say metaphase, think M for middle, because that's what happens in metaphase. The chromosomes line up in the middle. So that's a chromosome, right? And then there's another chromosome down there below it. For us, there'd be 46 of these chromosomes. This is a really simple cell with just 2 chromosomes. But they all line up in the middle of the cell, and the spindle fibers connect the sister chromatids to the centrosome. So I'm going to divide this one up. Here's spindle fibers. You ready? Spindle fibers, remember, we formed those during prophase. That's the fishing line. Okay. You haven't missed anything. Sister chromatids, remember, those are the 2 halves of the chromosome. So there's one, there's the other. So 2 sister chromatids, remember, they're identical to each other, semi-conservative replication, right? And then the last little bit, the centrosomes. Centrosomes are represented here by these circular guys. Again? Yes. Yeah, they look like Ks, I guess. Yeah, I think of them as X's, but yeah, they're Ks. Again, maybe just to blow them up. That's what they would be, right? So this is one sister, and this is the other sister. Okay? And they're identical to each other after DNA replication is when they form. Okay? So this is called metaphase, which we sometimes abbreviate with an M for metaphase. Now let's go to anaphase. Ana is a prefix that means after. And so this is after the sister chromatids get pulled apart and move to opposite ends of the cell. And so that's pretty much what we're seeing here. Nothing really new. Let's maybe just label the sister chromatids. There's one, right? There's the other, right? And so they got pulled apart. And now they're being pulled in by the centrosomes. I think of this as kind of like fishing. Anybody likes to fish, right? These centrosomes are kind of like fishermen. The spindle fibers, the strings, are like the fishing line, and that's pulling the chromosomes to the opposite ends of the cell. So literally half the DNA goes to the left, half the DNA goes to the right. For us, that's 46 chromosomes go to the left, 46 chromosomes go to the right. In this cell, it's just 2. Okay, and then the last part, telophase. Telophase. And telo is all about extremes, right? And so now we have 2 nuclei starting to reform at extremes of the cell. So let's go ahead and see this. This is sister chromatids are unwinding. That's pictured here. Oh, actually, maybe this would be a better spot. Let's do that. There we go. So the chromosomes are now unwinding, so that ball of string is going back to just being string again. Okay? The nucleus is going to reform. So I'm just showing that with a dotted line. And the spindle fibers, we don't need them anymore. So we take those apart. The spindle fibers fall apart. Everything that we're talking about here in telophase is the exact opposite of what other step? What other step was there when we took the nucleus apart? And second, not metaphase, good guess. And we wound the DNA into chromosomes. You guys remember? This is the opposite of prophase. So the first step and the last step are just opposites of each other. Everything that we did in prophase is now being undone. In prophase, the nucleus fell apart. In telophase, the nucleus reforms. In prophase, the DNA winds up into chromosomes. In telophase, the DNA unwinds back from chromosomes to the stream double helix. In prophase, the spindle fibers were forming, and in telophase, the spindle fibers are falling apart. The other thing that's noteworthy here, right, is how many nuclei do we have here? This is a weird moment in the cell because they have two nuclei. Right? It's one cell, but there's two nuclei. The nuclei are identical to each other. They have the same genetic code in them, but they're in the same cell still. Any questions here? All right. Can I take these annotations away, or you guys still seem like you're ready? Cover that we did. 9-inch nails. Hurts. Was it? Oh, hurts. Oh, that was so good. Oh, do you? You have a record player? Oh, that's impressive. Okay. We're not putting that in. That's right. Excuse me. Oh, no. We have one, couple more things to do. Sorry. Okay. I waited too long. Sorry. Okay, here we go. Okay, don't wait that long this time, Brandon says. Sorry, it's hard to read your body language. I can't tell if you're writing or you're just avoiding eye contact. All right, let's look here. This table is just a nice, simple summary of everything we talked about. Okay, prophase, metaphase, anaphase, telophase. Again, sometimes we think of this as PMAT. If that helps you remember the order of the steps, PMAT: prophase, metaphase, anaphase, telophase. And basically, we're just keeping track for all of these steps. What's going on with the nucleus, what's going on with the spindle fibers, and what's going on with the chromosomes. So in prophase, the nucleus dissolves or falls apart, the spindle fibers are forming, and the chromosomes coil up. In metaphase, the nucleus is gone, so it fell apart. Spindle fibers are parallel to each other, and the chromosomes line up in the middle. Anaphase, the nucleus is still gone, spindle fibers are pulling, chromosomes are splitting into the two parts. And then lastly, telophase, the nucleus is reforming, the spindle fibers are breaking down, and the chromosomes are unwinding, going back to the long point. Last step. Now the cell is finally ready to divide. It's got its DNA sorted out. It's got two nuclei. It just needs to divide. And so that's what's going to happen with cytokinesis, which sometimes we abbreviate with a C. Cyto means cell. And so in this case, the cell is going to divide. And what's basically going to happen is the cell membrane is going to pinch in the middle and form what's called a cleavage furrow. Okay, cleavage furrows. Let's just label that. Okay, the cell is pinching into two daughter cells right down the middle. It's really important where that cleavage furrow forms. We're now separating these two cells into two daughter cells. So eventually, if time goes on a little bit, this is going to be one cell, and this is going to be the other cell. Okay? Each with its own nucleus, each with its copy of the DNA. Now, in a plant cell, there's an extra step, because remember, plant cells are surrounded by cell walls. They don't just have cell membranes. So a plant cell, after it does cytokinesis, it has to form a new wall to separate the two daughter cells. So that's what the cell plate does. So at the end of this, you have two daughter cells. Okay. Any questions here? All right, that's all of the new information for today. If we could, let's go ahead and